بسم الله الرحمن الرحيم.
Assalamu alaikum students, hope you are well and you are enjoying the weather too. Today we continue the chapter 1 in the lecture number 3 for biology, introduction to biology. Chapter number 1, our third lecture is consisting of concepts of abiogenesis and biogenesis. As you remember, in the last class we have slightly discussed the topic. The concept of abiogenesis was presented by Aristotle, who was a Greek philosopher who has the name and value in the field of science, so he presents the concept that life came from non-life. Later on, an Italian biologist, Redi, performed some experiment, and in support of in light of his experiment, he suggested that life came from only life. So the concept or the theory he presented it was that living organisms are produced from the pre-existing living cells. In support of his concept, he performed some experiments that are known as Redi's experiments. So today we are going to discuss the Redi's experiment, Redi's first experiment. He performed three experiments in support of his concept. What does he do? He took a jar in which he placed some dead pieces or dead snakes. These dead snakes were placed in a jar, so the flies came there, visited on the dead pieces of snakes, and they lay eggs. And after that are invisible, the eggs of the flies are invisible. These eggs develop into the maggots or larvae, and later on these larvae developed into the flies that were the exact copy of the ancestor or the parents or the flies they visited earlier on the dead snakes. So he suggested that these flies are not the offspring of the dead snakes, while these are produced or these are the production of the ancestors flies that visited over the dead snakes. Later on, he performed another experiment that is known as Redi's second experiment. What was the Redi's second experiment? In his second experiment, Redi performed another experiment. He took 8 bottles in which he put the dead snakes and pieces of meat. Then he kept covered 4 of the jar while 4 remained uncovered. So first he took 8 jars and placed or put dead pieces of meat, snacks and some meat, and he kept open 4 jars and closed 4 jars. This time he do something different because in first experiment he do not cover the jar and he keep the dead snakes in the jar and the flies visited. So people said some of the believers of abiogenesis do not believe in the concept or in the experiment of Redi's. So they argue that the flies are produced due to the presence of air, as they believe in the concept of abiogenesis that life produced from non-life. So they start thinking that the flies are the offspring of the air, that enters into the jar, the dead pieces of snakes that are already present in the jar. So do not believe that the fly lay eggs, because the eggs were invisible. Then what happened in after placing 4 covered and 4 uncovered jar? So what happened after some days, after a few days, it happened that no maggots appeared in covered jar. Maggots were the larvae of the flies, and after a few days maggots appeared in the open jars. So this one is the evidence that fly cannot enter into the open closed jar. So they do not lay eggs in the closed jar, but when they enters into the open jar, they start laying eggs over the meat or the dead pieces of snakes. But later on, the believer of abiogenesis do not agree with the Reddy. They said that if air could enter into the jar, so the flies may produce from the dead pieces of meat, as they believe in the Aristotle that life is produced spontaneously from non-living organisms or non-living objects. So later on, Reddy performed another experiment, and what was the conclusion of this experiment? So he denied that air is not necessary to produce the life. He performed another experiment that is known as Reddy's third experiment. So what he has done, he placed a jar with the pieces of dead pieces of meat or dead snakes or fishes, This is how, in his third experiment, and covered the jar with a gauze, so through which the air can enter, but flies cannot reach to the meat. So what happened in the next? In the result of this experiment, no flies can go inside or remain inside. They remain outside the jar and they do not enter into the jar. Simply, in his first experiment, he do not cover the jar and he suggested that only flies can visit it, or the flies has visited the meat. That is why they feast on the meat and they lay eggs over there. But people do not believe in that. Believers of Aristotle, believers of abiogenesis do not believe in this concept or this experiment. They said that air can produce the flies cannot produce it. So he performed another experiment and he covered half the jar and keep uncovered remaining half jars. So the people still do not believe that. They said that if air could enters into the closed jar or open jar, so the flies can be produced by the help of the air. They do not believe in the experiment number 2 still. So he performed another experiment to kill the objection of the believers of abiogenesis. He put some pieces of meat in a jar and covered it with a gauze. A gauze is net. Okay? So he covered the jar with the net or a gauze. So the air could enter but flies remain outside. Again, no maggots appeared on the pieces of meat. Why? Because now he proved that air could enter into the jar and the flies would not. So the flies that were produced in the first experiment will definitely the offspring of the flies they visited over the meat. Basically, the concept of biogenesis and abiogenesis have an age distance and era distance between these two concepts. There were no experiment. Aristotle do not perform any experiment to prove his concept, while Redi performed some experiments to prove his concept. Redi performed a series of experiments. Later on, people still have some objections and some questions over the concept of biogenesis. Majority of the scientists start believing that life came from the living organism and the cells are produced from the pre-existing cells. So the living organism can only be produced by the living organism. But what happened next? Another scientist start performing experiment to prove the concept of abiogenesis. Here, Redi has performed series of experiments and convert his concept into a theory. While hypothesis that was presented by Aristotle, it is still remain hypothesis. So some of the scientists or that believe in the abiogenesis, or some of the scientists that want to prove the concept of abiogenesis start performing an experiment. So the Needham is the biologist who perform an experiment in support of abiogenesis. It was the first experiment he performed in support of abiogenesis. So, Needham is a biologist, is a scientist, English scientist who belongs to England. In 1748, he boiled some pieces of meat in water, poured the gravy into the jar, and closed the jar. After some time, in the closed jar, some of the microorganisms produced. He suggested that these microorganisms are produced by the water. Water is a non-living object. This concept or this experiment make them believe in abiogenesis, and this once again excited the believers of abiogenesis. They still believe in that life comes from the non-living. But later on, another biologist, Pasteur, performed an experiment. So we are going to finally discuss the last experiment of this concept, Pasteur's experiment. What does he do? He performed an experiment in support of biogenesis, again to make them believe in biogenesis. Louis Pasteur, a scientist, performed experiment. He took an S-shaped, a jar, a flask that is having a neck like this. Now this flask is containing a solution that is made up of sugar, yeast, and water. These all are non-living objects or non-living solution. So why he used this typical shaped flask? He performed an experiment by a different shape of flask to resolve the issue. He was a French biologist, and he performed this experiment in front of a board of scientists to resolve the issue of concept of biogenesis and abiogenesis. He boiled this solution, he heated this solution, and after some time he observed that no microorganism produced inside the jar or inside the flask, but the microorganism produced here in the curve of this S. This part may contain the microorganism. So he suggests that because of the typical shape of flask, the microorganism could not enter into it. So they are present in the air, and the microorganisms are carried by the air to the flask. So they are not produced by the solution that is present inside the flask. They are already present in the air. So they enter, when air enters into the flask through this opening, so it is stuck here due to the curve. So air could not enter into the flask, so microorganism also stuck here in this part of the flask, and they could not enter into the flask. So conclusion of today's class: Reddy and Pasteurs perform experiment in support of biogenesis, while neither perform an experiment to support abiogenesis. So abiogenesis remain a hypothesis that is not proved by any experiment, and the biogenesis is proved by the Reddy's experiment, and Pasteur performed another experiment in support of biogenesis. It is a theory that is proved by many experiments. So students, keep learning, keep studying. The next topic, inshallah, will continue the chapter 1 in the next class. Take care. Allah Hafiz.